Good afternoon, students. Today we are going to continue with the human reproduction chapter, male reproductive system. And in this video, we are going to talk about structure of a sperm cell. So in our last video, we spoke about spermatogenesis, and in this, we are going to speak about structure of a sperm cell. Now, this is the structure of a sperm cell, and we can see here that it is divided into head region, neck region, middle piece, and tail portion. So when we look into the head region, head consists of a big nucleus which is filled up with chromatin material. So the nucleus consists of chromatin material. Now, on the surface of this nucleus, it is surrounded by plasma membrane, and on top of this cell, the head, there is a cap-like structure which is called as acrosome. Now, this acrosome is a modification of Golgi body of the cell. Now, students, this is very important to be remembered because from here, many times question is asked in NEET about what is acrosome. Is it a lysosome or is it a Golgi body? Always everyone gets confused with this answer. It is always Golgi body modification and never a lysosome. But it consists of lysosomal enzymes. So Golgi body only give rise to lysosome, and all those lysosomal enzymes are found in this acrosome, and this lysosomal enzyme actually helps in fusion of this sperm cell with the egg cell during the process of fertilization. Now, we can also see that the neck region consists of two centriole. One is the proximal centriole and the distal centriole. Now, this proximal centriole helps in the first division of zygote. So, when that one single cell zygote starts its division, the spindle fiber formation is being aided with the help of this proximal centriole. Whereas the distal centriole function is still not very clear, apart from it continues as a axial filament. So it continues as a axial filament. And now when we look into the middle piece of this sperm cell, we can see that the axial filament is surrounded or wrapped by a sheath of mitochondria, and this sheath of mitochondria is also referred to as Nebenкern. Nebenкern is a spiral typical structure of the proximal tail region of the sperm. So, mitochondria here in the middle piece forms a helix-like structure, and so that mitochondrial helix-like structure is also referred to as Nebenкern, also found in certain insects, as for example Drosophila. So this is also found in case of Drosophila. And then we can also see this ring centriole found here, and the tail region which is surrounded by a fibrous sheath. So with this, we have understood little about the structure of the sperm cell. Now, 2012 NEET asked a question: which of the following is false regarding the viability of a sperm cell? Number one option was viability of a sperm is determined by its motility. Okay, so we have to understand which is the false regarding this. So this is true. Viability of sperm is determined by its mobility or its motility. Sperm must be concentrated in a thick suspension. It is also true. Number 3 says sperm is viable only for 24 hours. This is false. Why? Because sperm is viable for approximately 72 hours. And number 4th option, they said survival of sperms depends on pH of the medium. Okay, so this is again true because it is absolutely correct that if the pH of the surrounding of a sperm cell is changed, it can kill the sperm cell, and as a result, this statement is also true. So the option for this answer will be 3, and we can see we have come to the end of the structure of the sperm cell, and we are going to continue with our next topic in our next video.